Okay, I think we'll make a start. So, good afternoon, everybody. Welcome to our current webinar from our Industrial Biotechnology Innovation Catalyst. So, for those of you that don't know me, my name's Aileen Miller. I'm a professor here in the University of Manchester and leading on our place-based impact acceleration account grant, which is to create a Northwest ecosystem for biotechnology across the Northwest, led by Manchester and includes Liverpool region and also Cheshire. Includes all our higher education institutions within our region, and has got a strong industrial base across industrial biotechnology in all sectors, from pharma to materials, through to chemicals manufacturing and energy and agri-foods, etc. So very pleased to see everybody today, the week before Christmas, and James, I can already see, he's in festive mode with his jumper and his Christmas tree behind him. And I'm really pleased to introduce today's webinar. But before we kick off, just to remind everyone that we are recording the session today. If anybody doesn't want us to record the session or has got any issues with that, please reach out and let myself and or IBIC know. We'll take that into account afterwards, and really strongly encourage you to think about questioning. James and Pavitt are not on holiday yet, so really encourage them to create an interactive discussion as we move through the webinar. We'll have time at the end for a Q&A session. So if you've got a question, either pop your hand up, or if it occurs to you as we move through, please just pop it into the chat and we'll come to it at the end. So without any further ado, I'm very keen to introduce James Winterburn, who's a reader here within the University of Manchester in chemical engineering, and also his postdoc, Pavitt Wong-Surishot, who are both going to talk to you today about sustainability from Salinity. So I will leave it over to you, James, to talk about your research and how you're a great example of an academic who also dabbles in the commercialization side through his company, Holofirm. James, over to you.
Yeah, thank you, Aileen, for the introduction. And yeah, good afternoon, everyone. Thanks for joining this webinar that Pavitt and I are going to co-present. And we picked a catchy title, Sustainability from Salinity. So what we're going to focus on is the work that we've done over several years within the research group, all around how we can take halophiles, so salt-loving microorganisms, that's the salinity aspect, to make biopolymers, so biodegradable alternatives to kind of traditional petrochemical produced polymers. And that's the kind of sustainability part of this kind of two-part story around sustainability from salinity. So actually, Pavitt's going to present first and give an overview of the area and the sector. And then after he's done his bit, you'll hear from me again, and I'll talk about some of our kind of process engineering, bioprocess engineering type work that's been going on, and then a little bit about what the future holds. As Aileen said, encourage you to ask questions. Doing webinars is talking at my computer screen. So if you put something in the chat and I notice it as I go along, if it's pertinent, I'll address it, and if not, we can come to the questions at the end. Hopefully Pavitt and I won't get too carried away, and there'll be time for some discussion as well. So yeah, thanks for joining, and Pavitt, over to you.
Hi, thank you, James, and thank you everyone for joining us today for this webinar. So like James said, I'll be teeing him up for some talks later on in this showcase. But what I'll be doing first is to go through the background and set the scene for our webinar on sustainability from salinity, and what exactly is halophile PHA production and what is PHA. So let's start with that. So PHA, or polyhydroxyalkanoates, are a group of bio-based and biodegradable polyesters that are produced by microbes, so bacteria and archaea, and we'll be seeing both bacteria and archaea today. And they are stored as intercellular granules, so like those white granules you can see in the photo. And they are closely related to fatty acids. So their monomers are very similar in structure to fatty acids, and they are synthesized by the microbes by polymerizing fatty acids. And you can get all sorts from short chain to medium chain to long chain. Say short chain is, say, a 4 to 5 carbon or 6 carbon, like the poly 3 hydroxybutyrate covalent you see on the right. That's an example of a copolymer with 4 carbon and 5 carbon monomer. So that's a short chain PHA, and we'll be seeing a lot of short chain PHA today because a lot of halophiles like making them. But the important thing about PHA is that it's a notable candidate for as a sustainable polymer for commercialization. So it has the potential for a lot of applications in both traditional applications such as packaging and textiles. So you see some examples, some PHA-based products on in the pictures, but it also could have some novel applications. Like there's been research into using PHA for more sustainable electronics, and we'll be seeing some examples of PHA being used for the development of novel biomedical applications today as well. So that's a general background on PHA. And I said it's a good candidate for commercialization. And so we should have some idea of what the current commercial landscape of the PHA is like. And this will tie into how great halophiles can be later. So currently in the table, these are the big players or the major players in commercial PHA production. So some key points. There's not that many major players in PHA production. Their annual production capacity is relatively low, although there are announcements and commitments for expansion on the way, but this on the table is roughly what we have as of 2024. And there's a lot of PHA types I've listed here, but the essential gist of it is that the majority of the forms of PHA that have been commercialized are between 3 to 5 carbon. So this lies in the short chain side of the family. Now, this shows that the industry itself is still very nascent. So in comparison to something, let's say like polyethylene or PE, your big players are like Exxon and Dow and Sinopec, the companies that most people, even outside of the industry, have heard of. And this capacity for biopolymers is relatively small. So when we look at petroleum-based polymers, you're in the millions of tons per annum, and even petroleum-based ethanol is in the 100,000 of tons per annum. So there's obviously a lot of room to grow. And like I've said before, there's not that much commercialization of other types of PHA. So that's kind of where we are. And now there's this very nice paper from George Chen in Tsinghua, who's also involved with the commercial side of PHA production and his paper is on the grand challenges of industrializing PHA. So like what are the major areas that needs to be tackled to make PHA more mainstream, for example. So there's a lot of it, but I've broken it down into 4 main areas. So one is the cost. There's a lot of costs associated with making PHA, like the substrate costs, capital costs, because you've got to sterilize the process in biotech, and there's other associated costs like aeration and labor. That's also because it's a bioprocess, you've got your water-related challenges, and being a bioprocess, most of the work is done via batch fermentation. And there's also some other challenges in downstream processing. So there's some process-related challenges. And there's also productivity and product quality related challenges. So there's room to improve in terms of your product properties, product diversity, and improve PHA productivity in general. We should be making more PHA per gram, so more kilograms per liter per hour, essentially. So these are the major challenges and for commercial PHA production. And where the halophiles can come in is that a lot of these challenges can be helped by the use of halophiles. So like salt-loving microorganisms, they can help to provide more like novel solutions to addressing these challenges. And I'll go into more detail in the next slide. And combining the use of halophiles with something like genetic engineering, bioprocess engineering, and chemical engineering can help to address the other challenges and cover all of these grand challenges for PHA commercialization. So that's kind of the background as to why us in the Winterburn group and a lot of people within the PHA research scene have started to look into the development of halophiles with genetic and bioprocess and chemical engineering. So taking some of those challenges in green from the previous slide and through halophile production, halophile PHA production, we can see that halophile PHA production can offer some solution to these challenges. Like halophiles, because they're salt-loving bacteria, means that they can be grown at no or low sterility conditions because other contamination is less of a concern, since other microbes don't like the salt as much. So these will help with your capital costs because you don't have to worry about sterilization a lot. So your vessels doesn't have to be all stainless steel, for example. And again, there's no sterilization cost also is beneficial there. Halophiles production also can mean you want to be using salt water instead of fresh water, so that's reduce your freshwater demand. And because of the low sterility requirement, the process instead of batch processing, continuous process is more easily implemented with halophiles because there's less stringent sterility requirements, and this can aid with improving the overall productivity of the fermentation, and you can typically run continuous fermentation for longer, therefore you reduce your downtime. And with continuous fermentation, it helps, the use of continuous fermentation can help allow for better design for media recycling, and this can lead to reduced waste management and fresh water needs. Lastly, halophiles is also susceptible to osmotic shock because it likes salty water, but it doesn't like fresh water. So if you put your cell biomass with your polymer inside into fresh water, the water rushes in to the salty cells and it bursts. So this easier cell lysis of halophiles can aid in streamlining downstream processing and reducing the needs for more complicated cell lysis method. So that's the background of salty PHA production and why we want to be getting into salty PHA production. And today we'll be showcasing 4 projects within the Winterburn group that covers various aspects of how we can develop sustainable polymers production with salty microbes. So one is novel microbial organisms. So we do use organism called Halofrax quite a lot, which I'll go into more detail later. And James will also be talking about some of our more recent work with Vibrio for PHA production as well. And the key aspects of a lot of these works is process engineering, but also engineering biology that we work together with Neil Dixon's group in the MIB. And there's also sustainable feedstock to aid on the feedstock side of things that halophiles doesn't inherently improve. And there's also material science side as well, so to look at how the PHA made using halophiles can be used for more novel biomedical applications. And the 3 main areas that we focus on is improving the productivity, the composition, and the application of the PHA being made by the salt-loving halophiles. And it should help with cost and sustainability, like I've said before. So I'll set the scene with our first microbe is Halophorax mediterraneae, and this is a extremophile Heroarchaea. So it loves, like I said, it loves high salt environment, and it's pink, and it's very clear when everything's going well, is very pink. And when it's not going so well, you can see those flashes on the left. You can tell that your fermentation is doing well, and it makes PHA during the growth phase. And the type of PHA it makes is this polyhydroxybutyrate covariate, so P3HB co3HV, your PHBV, LC4 and C5 that I've talked about before. So this is the type that it likes to make as well. So I'll be talking through the first example, and then James will be taking you to the rest of the example in this showcase. So the first showcase is twofold. One is a showcase of how PHBV being produced by Halophorax have the potential to be used in novel tissue engineering as demonstrated by material science. And the other one is how we've applied sustainable feedstock for their production. So we've done some exploratory work for Halophorax made PHBV in tissue engineering, specifically towards ligament repair applications. Now, the good thing about PHBV or PHA in general, actually, for their application as tissue engineering materials like your scaffolds, that sort of thing, is because they're biocompatible. So you can stick them inside the body and they won't get rejected. They're biodegradable, so you stick them inside the body and over time they break down, which means that new tissue from the patient can start growing and replacing it. And if we process it right, and this is where the material science come in, they can have the correct or the suitable biomechanical properties for an intended application. In the case of this study, for ligament repair. And so what my colleague Tam has done is he's developed an electrospinning framework and conditions for different proportions of HV, which basically means that even though we call it PHBV and it's C4 and C5, you can have different percentage of C4 and C5 within the polymer. And the different proportion of C4 and C5 in the polymer changes its properties. So for example, having more C4 makes it more crystalline, having more C5 makes it more elastic, and that can then impact the type of applications and processing method that they can be used for. And so what Tom did was he developed this electrospinning method and look at the different mechanical, thermal, and surface properties of the fibers. This is where the material science come in. And we also look at the biocompatibility of cells on the fibers to see if they'll be suitable for tissue engineering. And so all in all, the bottom line is the cells grow very well on the fibers and they align in the proper direction that they should be, which means that the fibers, the PHBV fibers produced, have a high potential for being used for this type of tissue engineering. So this is kind of laying the groundwork for using salty microbes-derived PHA for this sort of application. Next, where Tom and I explored next is the use of sustainable feedstock for growing the cells and making the PHA. Because like you've seen in previous slide, the sustainable feedstock or the feedstock cost challenge isn't tackled directly by simply changing the producer. So what we've done is we've explored the use of rapeseed meal hydrolysis as a feedstock. So rapeseed is, well, all of us use rapeseed oil in our cooking, and half of the weight of the seed actually goes into the meal. So this is the rest of the seed after oil pressing, and it's an abundant relatively low cost and variable feedstock, making it a notable candidate for use in industrial biotechnology and biorefining, especially as a carbon and a nitrogen source. But it can also provide micronutrients for fermentation. So a lot of the time you can just hydrolyze the meal and a lot of microbes will just simply grow on the rapeseed meal hydrolysate without further supplementation. Now, rapeseed meal itself have been used widely in the industrial biotechnology research for making enzymes, antimicrobials, bioactive, surfactant, and PHA, and we've used rapeseed meal extensively in the group for making PHA. So, and lastly, rapeseed meal is of interest because if you can valorize rapeseed meal using biorefining, there's other contact point or potential integration point with other rapeseed related fractions like rapeseed straw or glycerol that is made from turning rapeseed oil into biodiesel. So it lends itself to what is called whole crop biorefining, where all of the fractions of the crop can go through a biorefinery process. So analogous to a petrochemical refinery process where all of the fraction is valorized. So that's just some background on rapeseed meal, which is the biomass that we like to use quite a lot. There's some more further reading if you are interested. So coming back to using rapeseed meal for HelloFrax, so what we did, Tam and I, was to see if the hydrolysate of rapeseed meal can be a viable fermentation media component for HelloFrax, because at the time, the majority of research into HelloFrax fermentation simply used synthetic media, so with stuff that comes from a bottle. So this was, if not the first, one of the first cases where a sustainable feedstock was applied to HelloFrax fermentation and PHA production. And we want to assess how well the rapeseed meal hydrolysate work as a media component. So as you can see from the diagram here, so the growth curve and everything, in gray and in red, you can see the cells grew really well on the rapeseed meal hydrolysate. This is at 75% rapeseed meal hydrolysate and 25% minimal synthetic medium. And the key point for this is that rapeseed meal hydrolysate works, and salty loving HelloFrax can grow on sustainable feedstock. And importantly, which wasn't really known before because most people typically use either glucose or fatty acids as carbon sources, that HelloFrax seems to be consuming a wider variety of sugar than expected, as you can see from the yellow line, which is galactose and cellulose coming down. And another important observation that we found was that the HelloFrax was making PHBV, which is the C4 and C5 PHA, without fatty acid precursors. Whereas in previous work, typically you'll see that organism would just make PHB, which is just C4 on sugars, and you'll need to be fed the C5 fatty acid, so valeric acid, as a precursor before it started accumulating PHBV. Now, with this experiment, it shows that with the right condition and right media, it can do that automatically. So there's some performance data that I'll quickly go through. Essentially, rapeseed meal can make up the majority of the media, and it works the best. So it shows that using sustainable rapeseed hydrolysate gives it potential to improve sustainability of the process, but also the performance of the process. And lastly, using rapeseed meal hydrolysate for PHBV production also change the properties of the PHA that is made. This mainly stems from the fact that the organism can automatically accumulate PHBV, and so the proportion of HV in the rapeseed meal derived pHa was actually higher. This led to a more elastic polymer compared to PHB that is commercially produced and PHBV that we've been making using synthetic media. So it shows that bringing in halophile production together with sustainable biorefining can actually improve the property of the material produced. Yeah, that's everything from me. So James will be taking over the next bit of the webinar. Thank you very much.
Yeah, perfect. Thank you, Pavit, for teeing me up there. So yeah, going to carry on the story now and kind of talk more broadly about some of the kind of process engineering challenges with producing these PHAs, polyhydroxyalkanoates, using halophiles. I noticed there are a couple of questions in the chat, so I'll pick up on those quickly. So Richard, the pigment is basically some sort of kind of carotenoid type molecule. We've never isolated or paid much attention to it other than kind of thinking about carbon balance and the fact that ultimately you wouldn't necessarily want lots of pigments being made. So there are some papers that look more specifically at these pigments, but it's not been the focus of our work, basically. The second question around do you need to supply a precursor or not, and it mentions ICI. You've teed up one of my little jokes quite nicely, so I'll get to that point in just a second. So thank you for that, Bob. So just on this idea of can we control the composition? So in the halophyrax, we're interested in making this copolymer that Pavit's already described, this PHBV. So kind of the background to a lot of what I'm going to talk about is some work that one of my PhD students did a little while ago now. And we were basically co-feeding volatile fatty acids, and the composition of the volatile fatty acid mix, so C4 and C5 volatile fatty acids, we basically showed that it's kind of the HBHV fraction in the polymer is proportional to that, which you can see in the figure on the right of the screen there. So this is the amount of C5, the mole percent of C5 volatile fatty acid in the mixture supplied against the fraction or the mole percentage of the copolymer that's HV. And so you get a nice straight line. So this is really great because this shows that in a process you can control the composition and actually the structure of the polymer as well, quite tightly, just based on how and when you feed these mixtures of volatile fatty acids. And basically you can get random copolymers if you co-feed. Sequentially feeding, you kind of get more blocky or blend copolymers. Pavit already spoke about the idea of incorporating a 3HV fraction increases elasticity, and this kind of gets around some of the problems of using pure PHB. So just to kind of give some insight into what these materials look like. So on the right is a PHBV film that we cast from some of the material that we made in the lab. And then getting back to this whole ICI thing. So the image on the left was actually sent to me by a professor at Stellenbosch University who in the 1980s, 1990s worked for a ICI subsidiary. And this is some of the material that ICI were producing back then. So my little joke that got set up there is that whatever you try and do in this industrial biotechnology space, ICI already had a crack at it at some stage. And that is actually largely true of a lot of things. And the interesting thing for me is actually, well, why didn't these things work, in the 80s or 90s? And there's some obvious answers to those things around kind of our understanding of sustainability and climate change and kind of what the motivations and drivers are. But I think it's important to see that whilst we're doing novel work, some of this is, goes back quite a long time. So yeah, the ICI biopol process, they were feeding precursors. We can do this with or without precursors, and obviously things like volatile fatty acids, you can imagine that you could get from an anaerobic digestion process, for instance, as a starting point, if we think about where we could get sustainable, potentially, feedstocks from. So from here, just a word on another thing that Pavitt mentioned, and that is kind of our mode of operation. So obviously, as chemical engineers, biochemical engineers, this is kind of bread and butter stuff, but it's worth thinking about why we might want to move from a batch to a fed batch to a continuous process. So what we have here is some of my little sketches of a batch process, a fed batch in the middle, and then a continuous process. And the main point here is that you get time variance in batch and fed batch processes. Obviously, in the fed batch process, we're introducing feed. So in general, we can get more, produce more product, more cells. But there's a limitation because we can only feed so much because the bioreactors is a given volume. That ties into capital expenditure and things like that. So the great thing about continuous fermentation is that over time, if you're running it properly, your biomass, your substrate concentration, and your product concentration are all constant. You can control from the feeding rate, which is related to your dilution rate, how quickly, the microorganism grows. So you get kind of direct control. You can dictate the behavior of the microorganism. And that's really useful when we're thinking about being able to have longer duration production campaigns, think about process control, having everything constant with respect to time simplifies things, especially at scale. We can also avoid inhibition from either substrate accumulation and having to have high substrate concentrations at the initial start of the batch fermentation, for instance. And also because we have, as well as feeding, we have an outlet from our continuous process where we're going to alleviate issues from kind of other molecules that might become inhibitory to the process as they're expressed during the fermentation. So what does this look like in this realm of producing biopolymers from halophiles? So we can kind of compare fed batch to continuous, and we've done a bit of work on this essentially at the kind of lab 3 litre scale. So just over a litre working volume at around 37 Celsius. So the point here is that we're working with the same types of conditions between the fed batch and the continuous, but obviously changing that mode of operation. So we can kind of have a look at what this kind of data looks like. So this is from kind of lab fermentations using Haloferix mediterranei, the time courses of these fermentations. So you've basically got your volatile fatty acid feeds in the squares. That's the total, and then the open triangles and the crosses are the components within that. You've then got your optical density, so something related to number of cells in blue, the circles, and then you can see the bars are the biopolymer, and the darker, shorter part of the bar is the 3HV fraction within there. And these are at 2 different carbon concentrations overall, so 0.1 molar in the top corner and 0.25 molar in the bottom corner. We also did this work at 0.4 molar because we were looking at the effect of this concentration on the fermentation process performance. So this is the concentration of carbon we're trying to maintain in the bioreactor. So our carbon from our volatile fatty acids, 0.4 is too high, as you can see. And then you can see some different things happening within these other 2 fermentations at these other 2 concentrations. So what we're really showing is that we can control effectively. And then if we look at some of these, again, these key metrics similar to those that Pavitt was talking about, what we're really interested in are our yield of product on substrate consumed. So that's our YPHA here in the second from left column. And then the final column is our productivity. So that's kind of really important too. And you'll notice here that we're at milligrams per litre per hour. So at the higher 0.25 molar carbon concentration, we get up to kind of 20 mg per litre per hour. And that compares favorably to this bottom row here, which is the batch case. So we can increase our productivity and we maintain, and this is key, we maintain the control over the composition of our polymer, which is obviously related to the material properties as well. So from there, we can go to continuous. So same type of data that I just showed you for the fed batch fermentations, but this time for continuous. And this second kind of pair of graphs here, these plots just show over time how much polymer we've actually produced, because obviously we're collecting the polymer from the cells that are coming and flowing out of the bioreactor. So we can, at these two different carbon concentrations, we can produce continuously and have good control over the fermentation overall and some sort of steady state. So again, if we look at the kind of fermentation data here, we can run these fermentations continuously. We get comparable productivities, so kind of 19 milligrams per liter per hour, and decent steady state production rates. And obviously the key advantage of a continuous process over a fed batch is that we can run for much longer duration. So I've not shown the data here, but I think that the longest duration fermentation of this type we've run was of the order of a month or so. So in terms of scaling up and our kind of scheduling, then in the course of a year, you can produce a lot more PHB because you don't have the downtime associated with having to stop your fed batch fermentation because you filled up your bioreactor and restarting and all of the kind of steps that includes. So really showing here kind of proof of concept of continuous fermentation with haloferrax. So one of the other things that Pavit mentioned when we was talking about some of the advantages or the potential to solve some of these common challenges around PHA production and the advantages of using halophiles is around kind of whether we can reuse components from the media and whether we can look at the effects of things like process temperature and other things on these fermentations. So we took the kind of best fermentation conditions from the continuous work at the relatively small scale and kind of scaled up to still kind of lab context, but up to kind of 8 liter working volume, similar conditions, kind of targeting a 0.25 molar carbon concentration in the bioreactor and feeding the same specific composition of C4, C5 volatile fatty acid mix within there. So from here, we actually ran these fermentations for a little longer than the data I just showed you. And the kind of key takeaway here is that at 37, we can see the behavior. The bottom left is now if we recycle the media. So what I mean by that is the salt. We've actually got more salt in the system than anything else in solution. So obviously that in terms of the process and kind of how you're going to deal with that effluent, the best thing to do is reuse that. So what we did was capture and collect the outlet from the bioreactor, remove the cells, harvest the PHA, harvest the cells, recover the PHA from them, and then that kind of salt solution that's left, use that as the basis for media to carry on feeding. So we add the volatile fatty acids and then carry on supplying that to these continuous fermentations. We also ran at 49 Celsius. So in general, when we think about a fermentation process, cooling is one of the key costs. So once you get to high cell densities, the heat energy that the cells kick out through their metabolic processes, we have to remove that. So actually cooling becomes a large cost and challenge potentially at scale. So if we can run at a higher temperature, we can reduce the operational costs of that process cooling. So reduce that cooling utility requirement. So if we kind of think about what we can kind of did here, the kind of data, again, so looking at yields and productivities, then we kind of do pretty well again here. So you can see reasonable yields, steady state production rate, and our steady state productivities here are higher, reduced slightly at 49 Celsius for basically you get much quicker growth there at the expense of biopolymer production. And then we can kind of look at whether this is kind of statistically significant in terms of the consumption rate of our volatile fatty acids. So going from a batch, a fed batch at 1.2 liters comparing to the 8 liter scenario as well. And really this is to say that whether we change the feed to being recycled, so that's B here, those fermentations are basically comparable in terms of their substrate consumption rate to the other conditions we tested. Things are very different when we change the temperature. And there's some reasons for that that I won't go into for now, but there's things that we could do there to further enhance the process. In the short amount of time I've got left, I'm going to touch on a couple of things relatively briefly. So looking at kind of engineering biology, strain engineering approaches, and then mention Vibrio nitrogens as a different halophile that we've also started to look at within the group. So very quick run through here. What we're interested in is, can we make other types of polymers, biopolymers in Halotherax? So we looked at making a terpolymer, so taking the PHBV and adding a CO4HB into the polymer structure. And there's several reasons for this, again, around material properties and kind of broadening the range of potential applications for these types of biopolymer materials. This is some work we did in collaboration with Neil Dixon in the MIB and Michaela Chacon, as well as Thorsten Allers at the University of Nottingham. And to kind of skip to the end of what we did, we basically selected some different 4-HB-CoA transferase or synthase genes and expressed those in Halotherax. And we're able to make reasonable amounts of the terpolymer of the 4-HB within the terpolymer, albeit with adding a gamma-butyl lactone GBL as a precursor. So we can express these within Halotherax, and then we took the Halotherax MCAT that performed better, and then actually used that to look at then how we can play with feeding in order to increase the production of the terpolymer within this fermentation system. So we can improve the terpolymer production, but we still have to look around these kind of common issues of toxicity or inhibition of the precursor that we're adding within there. We took this Halotherax an MCAT and used that as the kind of best performing strain to then look at in a kind of fed batch fermentation, and you can see kind of the time course of that here, and we're getting up to pushing kind of 0.8 of a gram of the terpolymer throughout this fermentation. So in the little bit of time I've got left, I'll just quickly introduce the idea of Vibrio nitrogens as a really interesting, in my opinion, organism for biopolymer production that comes with some of its own challenges as well. So this is a really interesting halophile for PHB production. It's incredibly quick growing. So this just kind of gives you a visual representation of that. Within a couple of hours of streaking on a plate, you can actually visibly see colonies. So it grows really quickly. The challenge here is meeting the biological oxygen demand and getting the glucose as the carbon substrate you supply to go to biopolymer and not through kind of other metabolic pathways into other molecules, other products such as gluconic acid. So kind of quite preliminary work here, but what we can show is that we can produce in a very short space of time kind of up to kind of gram quantities of PHB within a day, basically, or a little less than a day. So actually the peak PHB concentration we get here, we reach within kind of 10 hours. So I think there's a lot of potential here. So this is a kind of shot-wise addition, kind of fed batch fermentation time course. You can see the green bars of the biopolymer we're producing here, and then we can see the kind of process data from the bioreactor along the bottom here. So the takeaway from this is that we can get much higher productivity, so hundreds of milligrams as opposed to tens of milligrams, in the Vibrio compared to the halopharax. So this is one way of alleviating the kind of low productivity challenge. The thing here is, how do we meet biological oxygen demand and get these fermentations to run for longer? So kind of very promising preliminary data here, but there's a need for a lot more process engineering work to be done in order to really make use of the kind of high growth rate of this microorganism. So with that, I'll kind of bring the webinar to an end. So what Pavitt and I have been introducing is this idea of sustainability from salinity, using halophiles as PHA producers that can address directly some of these kind of commonly understood challenges and barriers to commercialization of industrial biotechnology processes for PHA production. We're interested in ultimately getting to high productivity, being linked to the economic viability of a process, having introduced kind of some ideas around novel biomedical applications, talking about material properties, and then kind of this combined kind of engineering at the microorganism level and the process engineering level to actually get to a viable overall process, with consideration of where we get the carbon that we're going to feed to the fermentation from as well, and what that might mean in terms of sustainability. It's kind of been a broad overview, so we've scratched the surface here. Not too much time to go into lots of detail, but in general, these kind of insights that were given around biopolymer production probably apply to kind of other halophile-based kind of biotech solutions as well. So with that, I'll finish up. I'd like to thank Pavitt for co-presenting with me. There's lots of people who contributed to the work. So in particular, Tam, who's at the front of the photo here, and Pavitt on the rapeseed and the material kind of biomedical work. Roger, who I think is actually on the webinar, did the Vibrio work, and then Sean Ray and Mariana did some of the other kind of halopharax work as well. So thank you for your attention, and happy to deal with any questions that we may have in the remaining time, if I didn't manage to filibuster and make it so there's no time for questions. But I think we have a few minutes, if I'm not mistaken. So thank you.
Yeah, brilliant. Thank you very much, James and Pavitt. Very insightful work there, going from production all the way through to some applications as well. If you've got a question and you're happy to show your face or hear your voice, please just pop your hand up in the chat. Put your hand up electronically, and I'll come along to you. But we've got a couple of questions in the chat still. One is from Scott Phillips. How is molecular weight of the growing PHA chain controlled during the production in the organism? And is this further processed by the commercial outfits to give a stable product?
Yeah, that's actually a very big question. So there's a lot of research, not necessarily done within our group, around how the microorganisms like halophiles actually build up the chains. And one of the challenges actually with biopolymers still, as I understand it, is molecular weight. And when you look at like polydispersity index, things that we want to look at as, if you're a polymer scientist, you're going to be interested in, slightly harder to control. And one of the things we have to get a handle on is that one thing we didn't mention: why do microorganisms make biopolymers? The primary function is a way of storing carbon. So they can, if there's a excess of carbon source, these microorganisms make biopolymers. They're storing carbon that they can then access later on, i.e. they can degrade these polymers, which is great. That very kind of mechanism is why they're biodegradable. The danger is, of course, that it's not as simple as the microorganisms make the polymer, and then if they run out of carbon, they then degrade it. It's actually a kind of continual process. There's kind of continued kind of recycling and like processing of the biopolymer. So if you don't get your fermentation right, essentially the microorganism degrades the polymer, which impacts molecular weight. So I'm not particularly answering the question, so I'll leave it there. But there's a need for consistent processes that control for composition, structure, and of course molecular weight, with the molecular weight being the tricky thing. There's things you can do like knocking out the genes responsible for degradation that get around that, and people do that sort of thing.
It'd be quite good to just, depending on the economics of it, to do like, to do some post production on the polymers coming out, to fractionate it down to molecular weights that you want for specific applications. But yeah.
Thanks very much, James, for that. I've got a few additional questions following on from that, but I might reach out to you by email.
Yeah, please do. Yeah. Cheers.
Brilliant. Got another question from Thomas Farmer. So how well does Helofrax perform in the absence of salt? Is it that it's salt tolerant, or is it actually performs better in the presence of high salt?
Yeah, it won't grow without the salt, is the short answer. So the salt is necessary. And we're talking 150 to 100 grams per litre kind of sodium chloride, kind of roughly. And other salts kind of concentration. So yeah, they have to be there. The microorganisms isolated from a salty lake somewhere in the Mediterranean, hence the Mediterranean part of the name. I forget exactly where, but yeah, without the salt, it won't grow. In fact, it'll just die. Which is why we use the osmotic shock that Pavitt mentioned as a kind of primary recovery step in the downstream. The cells just lyse and break open if you put them in tap water, essentially.
Okay, thanks, James. So I'm conscious of time, so I'm going to make this our last question, and it's from Stephen Minden. So given the rapid growth of Vibrio, do you foresee heat development a potential threat for large-scale, high cell density processes?
Not necessarily a threat. Obviously, some of the work I was mentioning was looking at operating at high temperatures to kind of reduce cooling costs. So I'm aware, like large scale, when you look at your techno-economics, that's going to be potentially a key area where you could reduce energy consumption and hence reduce cost of production. We've not thought about heat yet because we're one step back. So we're still looked at. So oxygen demand is kind of the key thing that is the first challenge to deal with. So fast growth rate, great. How do you make sure that growth isn't at the expense of production of polymer? And then how do you meet the biological oxygen demand? So we're still dealing with that kind of research challenge or research question, if you will. Temperature and other things are yet to come.
Perfect. And I'll read the last comment from Kirk. Nice presentation, Pavitt and James. And he noticed your sunglasses in that graduation picture, which was clearly the summer graduation at the end rather than the one that was here last week. So brilliant. Thank you very much, everybody, for hanging on. And thank you for those that are listening offline on our YouTube channel. So if you search the IBIC YouTube, you'll be able to find all our previous webinars available for you to view in your leisure. Very pleased that we've got our next webinar scheduled on the 23rd of January. So please feel free to join us for that. Click on the link on our web page to join that event, or look out for the adverts on LinkedIn. That webinar is going to be given by Femi Olaruniju, who's here today, and Alexandria Holland, both from Liverpool John Moores University, and they're going to be talking about repurposing serine integrases as tools for engineering biology. So thank you very much, everybody. Have a fabulous break over Christmas, and best wishes for the new year when it comes. And we will look forward to seeing you all in 2025. Thank you very much, everyone. Bye.